Gene editing stands as one of the most revolutionary scientific advancements of the 21st century, fundamentally reshaping our understanding of genetics, disease, agriculture, and even the very fabric of life itself. Its rise from the early days of imprecise genetic manipulation to today's precise programmable tools marks a journey of discovery, innovation, and ethical debate. The story of gene editing is not merely one of scientific progress, but also of societal transformation as the technology's potential to alter living organisms, including humans, raises profound questions about the future we are collectively building. At its core, gene editing refers to the deliberate alteration of an organism's DNA to achieve a desired outcome. This can involve inserting, deleting, or replacing specific genetic sequences, thereby enabling scientists to correct mutations, enhance traits, or introduce entirely new functions. The earliest attempts at genetic modification were cumbersome and unpredictable, relying on chemical mutagens or radiation to induce random mutations. These methods, though groundbreaking in their time, lacked precision and often produced unintended consequences. The advent of recombinant DNA technology in the 1970s marked a turning point, allowing for the targeted insertion of genes from one organism into another. This set the stage for the development of genetically modified crops and the production of therapeutic proteins such as insulin. However, it was not until the discovery and refinement of programmable nucleases, molecular scissors capable of cutting DNA at specific locations, that gene editing truly entered a new era. Tools such as zinc finger nucleases and TALENs provided greater specificity, but it was the emergence of CRISPR-Cas9 that catapulted gene editing into mainstream scientific and public consciousness. CRISPR, an acronym for Clustered Regularly Interspaced Short Palindromic Repeats, is a natural defense mechanism found in bacteria, which scientists repurposed into a powerful tool for editing genes with unprecedented ease and accuracy. By designing a short RNA sequence to guide the Cas9 enzyme to a precise location in the genome, researchers could induce targeted breaks in DNA, prompting the cell's repair machinery to introduce desired changes. The simplicity, versatility, and affordability of CRISPR have democratized gene editing, making it accessible to laboratories around the world. Unlike earlier technologies that required complex protein engineering for each new target, CRISPR can be reprogrammed simply by changing the guide RNA sequence. This breakthrough has unleashed a torrent of research, with applications spanning medicine, agriculture, environmental science, and beyond. In medicine, gene editing holds the promise of curing genetic diseases at their source. Conditions such as sickle cell anemia, cystic fibrosis, and muscular dystrophy, which are caused by single gene mutations, are now within reach of curative therapies. Clinical trials have demonstrated the feasibility of using CRISPR to correct faulty genes in patient cells, offering hope to millions affected by inherited disorders. Beyond monogenic diseases, gene editing is being explored as a strategy to combat complex conditions such as cancer, HIV, and cardiovascular disease. By modifying immune cells to better recognize and attack tumors, researchers are developing novel immunotherapies that harness the body's own defenses. In the case of HIV, gene editing has been used to disrupt the CCR5 gene, which encodes a receptor the virus uses to enter cells, potentially rendering individuals resistant to infection. The ability to make precise changes to the genome also opens the door to preventive interventions, such as lowering cholesterol levels by targeting genes involved in lipid metabolism. These advances are not without challenges, as the complexity of human biology means that unintended consequences, so-called off-target effects, must be carefully monitored and minimized. The potential of gene editing extends far beyond human health. In agriculture, the technology is being used to develop crops that are more resilient to pests, diseases, and environmental stresses. By editing genes that control flowering time, drought tolerance, or nutrient content, scientists are creating plants that can thrive in changing climates and provide improved nutrition. Unlike traditional genetic modification, which often involves the introduction of foreign DNA, gene editing can produce changes indistinguishable from those that occur naturally, blurring the line between conventional breeding and biotechnology. This has important implications for regulatory oversight and public acceptance, as some countries have chosen to exempt certain gene-edited crops from strict GMO regulations. Livestock breeding is another area where gene editing is making significant inroads. By introducing beneficial mutations or eliminating harmful ones, researchers are working to improve animal health, productivity, and welfare. For example, pigs have been edited to resist devastating viral diseases, while cattle have been engineered to lack horns, reducing the need for painful dehorning procedures. These advances not only enhance food security, but also address ethical concerns related to animal welfare and sustainable agriculture. Environmental applications of gene editing are equally promising. Scientists are exploring the use of gene drives, genetic systems that increase the likelihood of a particular gene being inherited, to control populations of disease-carrying insects such as mosquitoes. By spreading genes that render mosquitoes unable to transmit malaria or other pathogens, it may be possible to reduce the burden of infectious diseases on a global scale. Gene editing is also being used to engineer microbes that can break down pollutants, capture carbon dioxide, or produce biofuels, offering new tools for environmental remediation and climate change mitigation. Despite these remarkable achievements, gene editing remains a field in flux, shaped by ongoing scientific, ethical, and societal debates. One of the most pressing concerns is the potential for unintended consequences. While CRISPR and related technologies are highly specific, they are not infallible. Off-target effects, changes to DNA at sites other than the intended target, can have unpredictable and potentially harmful outcomes. Researchers are developing new variants of CRISPR, such as base editors and prime editors, which offer even greater precision and reduce the risk of unwanted mutations. Rigorous testing and validation are essential to ensure the safety and efficacy of gene-edited organisms, particularly when they are intended for clinical use or environmental release. The prospect of editing the human germline, making changes that are heritable and passed on to future generations, has sparked intense ethical debate. While somatic gene editing, which affects only the treated individual, is generally considered acceptable when used to treat serious diseases, germline editing raises profound questions about consent, equity, and the nature of human evolution. The birth of the first gene-edited babies in 2018, following an experiment in which embryos were modified to resist HIV, was widely condemned by the scientific community as premature and irresponsible. The event underscored the need for robust ethical frameworks, international consensus, and transparent public dialogue to guide the responsible use of gene editing technologies. Equity and access are central concerns as gene editing moves from the laboratory to the clinic and the marketplace. The cost and complexity of developing gene therapies may limit their availability to wealthy individuals or countries, exacerbating existing health disparities. Efforts are underway to streamline manufacturing, reduce costs, and develop scalable delivery systems, but ensuring that the benefits of gene editing are distributed fairly remains a significant challenge. Intellectual property issues, regulatory hurdles, and public perception all play a role in shaping the trajectory of the field. Public engagement and education are crucial to fostering informed debate and building trust in gene editing technologies, Misconceptions and fears about designer babies, genetic enhancement, and unintended consequences can fuel opposition and hinder progress. Transparent communication about the risks, benefits, and limitations of gene editing is essential to enable individuals and societies to make informed decisions about its use. Scientists, ethicists, policymakers, and the public must work together to establish guidelines that reflect shared values and priorities. The regulatory landscape for gene editing is complex and evolving. Different countries have adopted varying approaches to oversight, ranging from strict bans on germline editing to more permissive frameworks for research and clinical applications. International organizations are working to harmonize standards and promote responsible governance, but challenges remain in balancing innovation with safety and ethical considerations. The rapid pace of technological change means that regulations must be flexible and adaptive, capable of responding to new developments and unforeseen challenges. Looking to the future, the possibilities for gene editing are vast and expanding. Advances in delivery technologies, such as viral vectors, lipid nanoparticles, and physical methods like electroporation, are enabling more efficient and targeted editing of cells and tissues. The development of in vivo editing, making changes directly within the body rather than in isolated cells, holds promise for treating a wider range of diseases, including those affecting organs that are difficult to access or manipulate ex vivo. Researchers are also exploring the use of gene editing to engineer synthetic genomes, create novel organisms, and even resurrect extinct species, raising fascinating questions about the limits of biological engineering. The convergence of gene editing with other emerging technologies, such as artificial intelligence, synthetic biology, and regenerative medicine, is accelerating the pace of discovery and expanding the scope of what is possible. AI-driven analysis of genomic data is helping to identify new targets for editing and predict the outcomes of genetic changes. Synthetic biology is enabling the design of custom genetic circuits and pathways, while advances in stem cell biology are facilitating the development of gene-edited cell therapies for a wide range of conditions. The integration of these technologies is blurring the boundaries between natural and engineered life, opening new frontiers in medicine, agriculture, and environmental science. One of the most exciting areas of research is the development of gene editing therapies for complex, multifactorial diseases. While early efforts have focused on single gene disorders, scientists are now exploring ways to modulate multiple genes or pathways simultaneously, addressing the underlying causes of diseases such as cancer, neurodegenerative disorders, and autoimmune conditions. This requires a deep understanding of gene networks, regulatory elements, and the interplay between genetics and the environment. High-throughput screening, single-cell sequencing, and advanced computational modeling are providing the tools needed to unravel this complexity and design more effective interventions. The potential for gene editing to enhance human traits, such as intelligence, physical ability, or appearance, remains a topic of intense debate. While current technologies are far from enabling safe or predictable enhancement, the prospect raises important ethical questions about the definition of normalcy, the value of diversity, and the risks of exacerbating social inequalities. Most scientific and regulatory bodies have called for a cautious approach, focusing on therapeutic applications and prohibiting enhancement until a broader societal consensus can be reached. Gene editing is also transforming the field of reproductive medicine. Pre-implantation genetic diagnosis, combined with gene editing, could allow prospective parents to prevent the transmission of serious genetic diseases. However, this raises questions about the appropriate use of technology, the potential for eugenics, and the rights of future generations. Ongoing dialogue among scientists, clinicians, ethicists, and the public is essential to navigate these complex issues and ensure that advances in gene editing are used responsibly. In agriculture, gene editing is enabling the development of crops with enhanced nutritional profiles, reduced allergenicity, and improved resistance to pests and diseases. Scientists are editing genes involved in photosynthesis, nitrogen fixation, and stress tolerance to increase yields and reduce the need for chemical inputs. These advances have the potential to improve food security, reduce environmental impact, and support sustainable agriculture in the face of climate change. However, public acceptance and regulatory approval remain significant hurdles, as concerns about safety, environmental impact, and corporate control persist. Gene editing is also being used to combat zoonotic diseases, those that jump from animals to humans, by engineering livestock that are resistant to infection or incapable of transmitting pathogens. This approach could help prevent future pandemics and reduce the need for antibiotics in animal agriculture, addressing the growing threat of antimicrobial resistance. The ability to rapidly design and deploy gene-edited organisms in response to emerging threats is a powerful tool for public health and biosecurity. Environmental applications of gene editing are expanding rapidly. Scientists are engineering microbes to degrade plastics, sequester carbon, and remediate contaminated soils and water. Gene drives offer a potential solution to invasive species and vector-borne diseases, but their use raises concerns about ecological balance, unintended consequences, and the potential for misuse. Careful risk assessment, monitoring, and public engagement are essential to ensure that environmental applications of gene editing are safe and effective. As gene editing technologies become more accessible, the potential for do-it-yourself biology and citizen science is increasing. While this democratization of science can foster innovation and public engagement, it also raises concerns about biosecurity, oversight, and the potential for accidental or intentional misuse. Developing appropriate safeguards, education, and support for responsible research is critical to harnessing the benefits of grassroots innovation while minimizing risks. The economic impact of gene editing is significant and growing. The biotechnology industry is investing heavily in the development of gene editing therapies, agricultural products, and industrial applications. Startups and established companies alike are racing to bring new products to market, driving competition, innovation, and job creation. Intellectual property issues, including patents on gene editing technologies and edited organisms, are shaping the landscape of research and commercialization. Ensuring that the benefits of gene editing are shared broadly, rather than concentrated among a few companies or countries, is an ongoing challenge. Education and workforce development are essential to support the growth of the gene editing field. Training the next generation of scientists, clinicians, regulators, and ethicists requires interdisciplinary curricula that integrate biology, engineering, ethics, law, and public policy. Public education initiatives can help demystify gene editing, foster informed debate, and build trust in science. Outreach to underrepresented communities and countries is important to ensure that the benefits of gene editing are accessible to all. Looking ahead, the future of gene editing will be shaped by ongoing scientific discovery, technological innovation, and societal debate. Advances in precision, efficiency, and delivery will expand the range of applications and improve safety. Ethical frameworks, regulatory oversight, and public engagement will guide the responsible use of gene editing, balancing innovation with caution. The integration of gene editing with other emerging technologies, will create new opportunities and challenges, requiring ongoing collaboration and dialogue among stakeholders. Gene editing has the potential to transform medicine, agriculture, and the environment, offering solutions to some of the most pressing challenges of our time. Its power to cure disease, improve food security, and restore ecosystems is matched by the responsibility to use it wisely and equitably. The choices we make today will shape the future of gene editing and its impact on society for generations to come. As we stand at the threshold of a new era in biology, the promise and peril of gene editing demand thoughtful consideration, open dialogue, and a commitment to the common good. In conclusion, gene editing represents a profound leap forward in our ability to understand and shape the living world. Its journey from the laboratory to the clinic, the farm, and the environment is marked by both triumphs and challenges. The technology's potential to cure disease, enhance crops, protect the environment, and even reshape human evolution is matched by the ethical, social, and regulatory questions it raises. The future of gene editing will depend not only on scientific and technological advances, but also on our collective wisdom, values, and vision for the kind of world we wish to create. As we navigate this uncharted territory, the story of gene editing will be written not just in the language of DNA, but in the choices and commitments of humanity itself.